It's Michael. Good morning, everyone. So untuk kelas hari ini, okay, we're going to discuss about cell fractionation as well as microscope, right? So the microscope involves the light microscope as well as the electron microscope. Okay. So now cell fractionation and differential centrifugation. Okay. So this is the centrifugation where your specimen is being spun in high speed, right, to separate their components. So when it comes to components of cells, cells consist of many organelles, right, as well as subcellular components. Organelles dengan subcellular components. Subcellular components macam microtubules, intermediate filaments. But we want to separate this from the cells. So one way is we use centrifugation. All right. So now cell fractionation is a technique, right, for studying cell structure and function, which takes cells apart and separates major organelles macam mitochondria, apa lagi, the nucleus, okay, ribosomes, and other subcellular structures from one another by using centrifuge, right? Centrifuge is sort of like a square, like a machine, right, where we insert this centrifuge tube, and then this machine will spin, all right, spin the centrifuge tube, and that will separate our cells components, right? So cell components are separated by size and density. Okay, size and density. The centrifuge utilizes heavy gravitational force to spin down the cell components of different S values step by step. As you can see here, the cell components are being separated, all right, step by step, all right, by their size and density based on the different S value or sedimentation units. The centrifuge, all right, will spin the test tube or centrifuge tube, all right, holding the mixtures of disrupted cells at a series of increasing speeds. All right, now cells, okay, we take an example, tissue cells, all right, we homogenize them, meaning to say kita hancurkan dia, right? So when these tissue cells are already hancur, the hancur we refer as homogenate. Okay, this is a homogenate. Okay, satu campuran di mana cells, tissue cells tu sudah dia hancur. Alright, not completely destroyed. It's just that the cell wall for plant cells, the plasma membrane for tissue cells, they already broken down. When these cell walls or plasma membrane of cells are broken down, what happens? The organelles will leave the cells, right? Now, at each speed of this differential centrifugation, right, a subset of cell components will settle at the bottom of the tube to form pellet. This is pellets, right? Yang macam solution ni ialah supernatant. So, boleh ya? Okay. Now, the supernatant, which is above the pellet, okay, yang ni pellet, ni supernatant, will be poured into the next tube for the next differential centrifugation. So, meaning to say, dalam centrifuge tube ini, kita ada homogenate, alright? So, centrifuge akan spin homogenate ni. Alright, spin dan pisahkan komponen di dalam homogenate ni. So, the first spin, alright, or the first differential centrifugation, we will get a supernatant and pellet, alright, and then the supernatant we pour into another centrifugation tube, and then we centrifuge that tube, pusing lagi, right, dan kita dapat lagi pellet and supernatant. Dan supernatant kita letak lagi dalam centrifuge tube yang lain, and then we centrifuge by differential centrifugation. proses memusing ni dipanggil differential centrifugation, right? A series of increasing speeds. Now, let's further discuss about this, the outcome of this differential centrifugation. Right, at lower speeds, which is here, all right, centrifuge at 1000 G. G means force of gravity. Right, so at 1000 um, G, at 1000 G, right, which is at lower speed, all right, or the lowest speed. So the pellet will consist of larger and heavier or denser components, and usually the nuclei and cellular debris will settle, okay, as the pellet at the lowest speed, or at 1000 G, right? Okay. Subsequent at 20,000 G, right? We will find right mitochondria and chloroplasts if we use plant cells in the pellet. And this supernatant we put into another centrifuge tube, and then we spin or let it undergoes differential centrifugation at 80,000 G. Okay, the pellet will be rich in microsomes, which is pieces of plasma membranes and internal membranes. Right, now the supernatant we spin again at highest speed, which is 150,000 G. So the pellet that we get, okay, we will observe, we will expect to get ribosomes. So here we can conclude that ribosomes has the smallest, okay, or lighter or the lesser components, right? Because higher speeds result in a pellet with smaller and lighter or less dense components. So finally, purification may be done through equilibrium sedimentation, okay, and the desired layer is extracted for further analysis. So meaning to say, all right, now for example like this, right, this pellet here at the lowest speed, lowest differential centrifugation, we have nuclei and cellular debris. So how do we purify this nuclei from the mixture? So we do equilibrium sedimentation, right? The same goes here as well, right? We have mitochondria and chloroplast, for example. So how do we separate these two? How do we purify the mitochondria, or how do we purify the chloroplast? So we do equilibrium sedimentation. Okay. Now, when it comes to S value or sedimentation units, all right, heavier molecules has higher S value. So we can see that nuclei has bigger S value compared with ribosomes. Okay, so summary, you are expect to see ribosomes, all right, as the latest component, whereas the nuclei, right, as the heaviest component or largest component. Okay, now light microscope, right? They are microscope, all right, that uses light to form images. Okay, and then light microscope uses two magnifying lenses, right, to achieve very high magnification and clarity. Okay, so the first lens will focus the image of the object on the second lens, which magnifies it again and focuses it on the back of the eye. Right, so there are many types. Okay, two types for your level SDPM candidates: compound microscopes and phase contrast microscopes. So this is what I believe most of you have seen in your lab before. This is the compound microscopes. So it contains objective lens. Okay, now when it comes to the structure of this light microscope, okay, candidates, these are the objective lenses, right? You can see, right? Objective lenses. The smallest will be 4x or 4 times. The medium will be 10 times or 40 times, right? And the highest or largest objective lens would be either 100 or 40 times or 100 times. It depends on the microscopes. But for in our lab, okay, our microscopes are based on this. All right, lowest we have 4 times, medium we have 10, and the highest we have is 40 times. Right. This is the mirror, right, where we focus the light into the object, specimen. This is the stage where we place our specimen. Okay, and this is the objective lenses that I mentioned earlier, and this is the eyepiece. Now class, in this eyepiece, right, there is another lens here, which is uh, known as ocular lens. This ocular lens, they are always magnify, all right, or comes with power of 10 times. Okay, 10 times. And then if you notice, there are two knobs here. This is called the fine focus knob. This is the coarse focus knob. Right? So when you place your specimen on the stage, okay, we first will adjust the coarse focus knob to get a glimpse or a faint image of the specimen. Then to sharpen the image, all right, of the specimen, we use the fine focus knob. All right. Now, this is a fast contrast microscope. Okay, a fast contrast microscope is a compound microscope that has the annular diaphragm or annular ring here. Okay, which is just under or below the stage, right? And then a face plate, okay, a face plate which is above the stage that form cone of light and change the background light of the image on the specimen. Now, three important parameters in microscopy, okay, magnification, resolution, and contrast. So magnification is the ratio of an object's image size to its real size. Light microscopes can magnify effectively to about 1,000 times the actual size of the specimen, right? At greater magnifications, additional details cannot be seen clearly. Dia jadi kabur, right? You will learn this in detail during our practical. Okay, your first experiment will involve usage of light microscopes. So you will deal, okay, with this magnification in more depth later on, hands-on, right? Resolution is a measure of the clarity of the image. It is the minimum distance between 2 points. Sorry, it is the minimum distance 2 points can be separated and still be distinguished as separate points. So light microscope cannot resolve detail finer than about 0.2 micrometer or 200 nanometers, regardless of the magnification. So meaning to say, right, mana yang lebih kecil daripada 0.2 micrometer, dia boleh guna light microscope. It's pointless. Okay, because you won't be able to see, to distinguish, okay, the image. The resolution dia akan lari, something like that. Next we have contrast, which is the difference in brightness between the light and dark areas of an image. Methods for enhancing contrast include staining or labeling cell components to stand out visually. Okay, contrast ni, either you want the image to stand out or you want the background of the image to stand out. That is contrasting. Okay, usually contrast we don't apply in Form 6, right? Maybe in the uni kamu akan apply dalam eksperimen kamu contrast ni. Okay, now this is a compound image that formed by compound light microscope, whereas you have this image that can be produced by a fast contrast light microscope. Boleh nampak kan? You see how the background is being darker and the specimen is being brighter. Or you can make the specimen darker, the background brighter, right? Nampak beza dia. Alright, electron microscopes. Okay, so electron microscope uses electron beams, right, that has 1,000 times the resolving power of a light microscope. So I mean to say, electron microscope can magnify way better in terms of resolution and magnification compared with the light microscope. Okay, there are two types of electron microscope. Let's go to the first one first, which is like in this diagram here. This is a transmission electron microscope. Usually it is being referred as TEM. Okay, TEM, transmission electron microscope. But again, the resolution is limited to 0.2 nanometer. So this can be used to study the internal structure of the cell. Maksudnya dalam sel tu kita boleh tengok apa isi dalam sel itu. Alright. So in this TEM, right, uh, electron beam, okay, is being aimed through a very thin section of the specimen. Okay, the specimen must be stained with atoms of heavy metals, which attach to certain cellular structure, thus enhancing the electron density of some parts of the cell more than others. Boleh faham kan maksud dia? Specimen tu kena stain dengan heavy metal. So bila specimen tu, alright, dalam specimen tu, we assume, let's say, a plant cell, alright. Some of the cellular structure, okay, plant cell tu dia akan melekat dengan heavy metal ni. So dia melekat dengan heavy metal tu, mana kawasan yang dia melekat dengan heavy metal tu kita boleh tengok dan lebih jelas. Bila elektron tu dia directed, right, at those structure. So the electrons passing through the specimen are scattered more in the denser region, so fewer are transmitted. Okay, so the image displays the pattern of transmitted electron. So both the SEM and TEM use electromagnets as lenses to bend the paths of the electrons, ultimately focusing the image onto a monitor for viewing. Okay, SEM is scanning electron microscope. Okay, when it comes to SEM, all right, it can't focus the detail of the organelles or the inside of the cell, but we can have a 3D view, right, of the cell. Itu beza SEM dengan TEM. Okay, scanning electron microscope seperti dalam gambar ini, nampak beza dia. Alright, useful for detailed study of the topography, meaning to say the external view of a specimen. So electrons are being beamed onto the surface of the specimen, right? So the electrons will be reflected back from the surface of the specimen together with other electrons that the specimen itself emits as a result of the bombardment. Bombardment means ditembak. So elektron tu akan tembak di permukaan spesimen, right? Dan elektron yang ditembak tu akan pantul balik. So kesan daripada tembakan tu, alright, elektron tu dipantulkan balik, kan? Kemudian the electrons tu amplified and transmitted to a screen where the image can be viewed and photographed. Can you see here, right? It's being viewed and photographed. Scanning electron microscopy yields striking 3D images. So, alright, so ini mesti SEM because you can see the 3D image of the topography of the specimen. Cantik kan? Tapi yang ni pula nampak dalam dia. Sangat jelas. So, this must be TEM, Transmission Electron Microscope. So, boleh beza gambar aja kalau keluar dalam exam. Which one is SEM, which one is TEM. SEM nampak 3D, TEM nampak dalaman. Alright, again, this is the SEM, Scanning Electron Microscope output. And this would be the TEM. You can see the organelles inside, very clear. Alright, this is SEM. Okay, so now, for your level, you must be able to differentiate between these three microscopes. The light microscope with the transmission and the scanning electron microscope. Alright. So, these three microscopes, okay, are focused by similar principle, which is a beam of light or electron beam is directed at the condenser lens. You can see the condenser lens here, right, for the light microscope, and the condenser lens here for the transmission and the scanning electron. Scanning electron adalah dua condenser lens. Okay. A beam of light or an electron beam is directed by the condenser lens onto the specimen, right? And is magnified by the objective lens. That's why we have the objective lens. Okay, even in this electron microscope, so they have the objective lens. Okay. But in the scanning electron, they don't have objective lens, but they have the projector lens. Mana projector lens? Okay, projector lens, not shown here, right? They have a projector lens here, all right? That um, magnified, all right, the electron beam. So, the transmission electron image is focused onto a fluorescent screen, and the SAM image is viewed on a type of TV, right? Television screen. Can you see the difference here? Okay, the image from the TEM will be focused onto a fluorescent screen, but for SEM, you can view it on a TV, right? Television screen. So the lenses in electron microscopes are actually electromagnets that bend the beam of electrons. Okay, so that's it for today.